All right, so the next topic here is preserving those cultures that we grow in those petri dishes. And of course, the best way to do it is refrigeration. So the funny thing is when I took microbiology at UD and we would put our petri dishes in the incubator because you have to incubate them at human body temperature for 24 to 48 hours, we actually had to go back to the lab on our own time and do everything ourselves, like take them out, collect results, dispose of them properly, whatever. Well, that's because most people at UD live on campus and they can kind of get away with that. We don't do that at Deltec. All right, so what would happen is, when we do the lab, so we would do the lab on a Tuesday, I would put them in the incubator and everything. And then 24 hours later, I would go back and I'll see a lot of times see if we done Tuesday night and I wouldn't be at work Wednesday night. So a lot of times I would do it Thursday morning. So it would be more like maybe 32 hours, but it's fine. It would be fine. So I would take them out of the incubator and put them in the fridge. And then they would stay in the refrigerator until we had lab again. And then you would get your petri dish, check your results, do whatever else you had to do with it. And that's how we would do our lab. So luckily, refrigerating the cultures after they have grown in the petri dish works very well. Now, of course, freezing them would work longer term, but a lot of times when you freeze them, it could kill some of your growth, so you're not going to get as good of results, and it can also definitely affect the media in the petri dish. Freeze drying works as well, but we don't have that complicated equipment at Deltec, so definitely not something we would be doing. And then you can actually keep the cultures, frozen for a very long time, and then they can be thawed out, and they usually come back to life, which is a little bit scary if you think about it. But yeah, bacteria have been doing that for a very long time. They can survive being frozen and then being thawed back out, which is why learning about the danger zone is so important. All right, so we know that bacteria reproduce in a process called binary fission. Some bacteria can do what mold does, which is called budding. We're not going to worry about that, though. Just stick with binary fission, keep it simple, and we'll be good. And of course, this is considered to be asexual reproduction because essentially the bacteria is just cloning itself or doubling itself. It's binary fission is basically a very simplified version of mitosis. That's all it is. So generation time is the time required for the population to double. So lots of bacteria, the generation time is measured in minutes. And because bacterial growth is so fast, the growth curve is actually a logarithmic curve. Now, don't worry about the term logarithmic. I'm sure that's a little bit of a scary word for a lot of you. All it really means is everything is by powers of 10. So instead of things being, 1, 2, 3, 4, it's 1, 10, 100, 1,000, 10,000. So powers of 10. That's what it means when we say a logarithmic scale. Now, if you want to look in your book on pages 164 and 165, it talks more about that. It shows you the math involved, all that kind of cool, fun stuff. We're just not going to worry about it for our purposes, okay? So here's a picture of binary fission. Now, you're not going to be labeling this or drawing this, nothing crazy like that. It's just to kind of show you how it happens. Very, very similar to mitosis, okay? It's just how bacteria does it instead of eukaryotic cells. All right, so in terms of the bacterial growth curve, you do need to know the phases, so you want to know these definitions. The lag phase is the gearing up phase where cells are active and basically preparing to divide. Then you have the log phase, which is also known as the exponential growth phase. This is when the bacteria double or grow. So, if there's 10 bacteria cells, 10 become 20, 20 become 40, and they just double, 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 double. The stationary phase is when growth ceases, usually due to the depletion of nutrients, the buildup of waste, the buildup of acids, that kind of stuff. Remember, we're talking about the growth of bacteria in a petri dish, not like, in a lake or something like that, where they may never reach the stationary phase unless, the lake freezes over in winter or something like that. And then finally, the death phase, that's when the bacteria die. So treat these 4 terms as vocabulary words. You also want to know them in the correct order. So lag, log, stationary, death. And then the other thing you need to be able to do is label this figure. And it's pretty easy if you know them in the right order, right? So again, it'll be multiple choice. You will be labeling the parts of this diagram. But again, if you just remember lag phase, log phase, stationary phase, death phase, you're good. So if you know them in the right order, you should be able to label this pretty easily, I hope. Okay, so now in order to measure how much bacteria actually grew, we do something called a plate count, which means you're going to count the number of colonies. But in order to do this, you have to dilute out the sample. And I'm going to show you what I mean by that in a second. And then we're also pour plate method, spread plate method. We'll talk about that in a minute as well. Okay. So let's first talk about serial dilutions. Okay. Because in a sample, there is so much bacteria that you have to dilute it out in order to be able, first of all, in order just to be able to count it, but then to also get an accurate answer, okay? So basically, we have our original, it says original inoculum, okay, so original sample, original culture, whatever you want to call it, and then you take 1 ml from that original sample and you put it in 9 mLs of broth. Then you kind of mix that one. You take 1 ml from this one, put it in another one, mix that one, do it again, do it again, do it again. And that's why we call this a serial dilution. So this tube is a 1 to 10 dilution. This one is 1 to 100, 1 to 1,000, 10,000, 100,000, okay? Then what you do is you take a little pipette and you take 1 ml from each test tube and you put that 1 ml on a petri dish, and you're either going to do this with pour plate or spread plate, which we'll again talk about here in a second, all right? And then basically you let the bacteria grow and you see what happens, right? So you got to put it in that incubator, let it incubate for a day or two, bring it back out, and then you take a look at it, all right? Well, looking at this one and looking at this one and looking at this one, right? All the pink is the bacteria. There is no way that you'd be able to count how many colonies are in these first 3. There's just way too many. It's just way, way too much. Now for this one, you can definitely count these. And it'll take you a little bit of time, but not a lot of time, right? But then what about this 5th one? I mean, this is like 1, 2, 3, 4. Like that you can do in a second, right? So let's say you're working for some company and your boss says, well, I need to know how much bacteria was in there right now. Like I need an answer immediately, as soon as you can give it to me. Well, you can say, all right, well, 1, 2, 3, 4. 4 times 100,000, because it's a 1 to 100,000 dilution. You could say, okay, there were 400,000 bacteria in that sample. And you're like, okay, whatever. That's the answer. That's the final answer, right? But if you want to be more accurate, if you want to be more scientifically correct, you should do this one. If you were to count all of these individual colonies, right? 1, 2, 3, 4, 5, right? You would come up with 32. There are 32 individual colonies on the 1 to 10,000 dilution. And 32 times 10,000 is 320,000. 320,000 is the amount of bacteria in the original sample. That, estimate is much more accurate, okay? Now, if you need a much quicker, faster answer, you could go with the fifth one. 400,000 compared to 320,000, it's not too bad. But you want to use the Petri dish that has the most amount of growth that you can actually still count. So the fourth one is the best one to actually use when you estimate how much was in the original sample. So here's pour plate and spread plate method. And again, this is how you would take the 1 ml from your tube and put it on the Petri dish in order to figure out the growth. Pour plate is you start out with an empty Petri dish, you put your sample of bacteria on it, you add the melted nutrient agar, swirl it, and then let it solidify and grow. The problem with pour plate is, one, the melted agar is kind of hot. The bacteria might not appreciate you pouring this hot stuff on it. I mean, it's not like boiling hot, but it's still pretty hot. You could also splatter a little bit on you, and you get little pinprick burns on your hand, which we've all done that when you do it yourself. Plus, the colonies kind of grow all throughout the agar, and it's harder to really count. It's harder to see them because some of them are on top, some of them are embedded within. I think the spread plate method is much, much better, and this is the more accurate one. This is the one that most places do. Pour plate is just kind of why would you do that? I mean, unless you're comparing the two, that might be kind of a fun experiment, okay? So spread plate is you have a Petri dish that already has the media on it. You add your sample, you spread it around evenly, let it incubate, and then you can count your colonies very easily because they're all just on the surface. They're not embedded within there. So spread plate, definitely the better method for sure. Okay, other techniques for measuring bacterial growth, filtration, so you can put it through a filter. Most probable number is a statistical estimation. A direct count, where you actually have to use a special cell counter. Nowadays, there's computers and technology that can figure this out for you, so that's kind of nice. Turbidity is cloudiness, which requires the use of a spectrophotometer. A spectrophotometer, it measures how cloudy a liquid is, and then you can basically extrapolate from there and say, okay, well, the cloudier it is, the more bacteria is in there. Measuring the metabolic output, such as measuring the amount of oxygen or the amount of CO2 that's given off by the bacteria. Dry weight, which is really better for fungal cultures. And that's it. I sped through those last two because honestly, I'm not going to ask you that stuff on the test. The most important was just the stuff about growing the cultures and pour plate, spread plate, and understanding, that the method of counting the colonies on the Petri dish. That's the most important. The other stuff I wouldn't worry too much about. I mean, just know what's on the PowerPoint. Maybe it'll be one question, but again, not our focus. So we're not going to worry too much about that. All right, that's it for chapter 6.